Hello viewers. Today my lecture topic is on protein metabolism. Now before going into the finer details of protein metabolism, I will give you a brief overview on metabolism. Now what is metabolism? Metabolism is a highly integrated network of chemical reactions catalyzed by allosteric enzymes. It can also be defined as coupled pathway consisting of both exergonic and endergonic processes. Metabolism proceeds through various intermediates, hence it is also known as intermediary metabolism, and the reactants, products and the intermediates are collectively called metabolites. Now here we can see that how the different fuels, proteins, carbohydrates and lipids are metabolized, and they ultimately converge into a common intermediate called acetyl-CoA. Acetyl-CoA is then further metabolized to carbon dioxide and water, and the free energy released is conserved in the form of ATP, which is the energy currency of the cell. Now there are four principal characteristics of metabolism: irreversibility, the presence of an exergonic first committed step, strict regulation at that step, and compartmentalization. Now metabolism can be categorized into two pathways. One is catabolic pathway and another is anabolic pathway. Now what is catabolism? Catabolism means fuels are degraded into simpler units and it proceeds through three stages. Now here in the diagram we can see the three stages of catabolism. The triglycerides, glycogen and proteins are first broken down into simpler units like fatty acids, glucose, amino acids. These are then further degraded into still simpler intermediates. One of them is pyruvic acid, another is acetyl-CoA. These two are then converted to carbon dioxide and water through the intermediacy of TCA cycle, electron transport system and oxidative phosphorylation, whereas the free energy released is conserved in the form of ATP. The biosynthetic pathway is known as anabolism. Similarly, like catabolism, it also proceeds in three stages. But here, the precursor molecules generated in catabolic pathway are first converted to building blocks which are finally assembled into macromolecules. Now to summarize the two pathways, here we can see in the diagram that complex metabolites are degraded into simpler products, and the free energy is conserved in the form of ATP and NADPH, which in turn provides the free energy for biosynthesis of various macromolecules. Now here in this diagram we can see how the catabolic and anabolic pathway takes place in reverse order and how these are interconnected. Catabolism and anabolism takes place, before I have said this, that they are compartmentalized, so they are distinct and separate and are simultaneously and independently regulated. But even then, there is a common central route known as amphibolic pathway which has a dual function. The word amphibolic means it can go either towards catabolism or towards anabolism. Now, after giving this brief overview of metabolism, I will now finally switch over to protein metabolism, which is my main topic of today. Now, as we all know, proteins are polymers of amino acids. Now, the sources of amino acids may come from two ways. First is the digestion of proteins, resulting in the formation of exogenous amino acids. Then is the degradation of unwanted polyubiquitinated damaged proteins through proteasome degraded pathway, which results in the formation of endogenous amino acids. Now amino acids, as we can see in this diagram, not only serve as protein monomeric units, it has multifarious roles. It also serves as energy metabolites in the synthesis of various important biomolecules like tissue protein, plasma protein, different antibodies, hemoglobin, glucose, glycogen, lipids, non-protein compounds like heme, heterocyclic amines, etc. Amino acids can be classified into two major types. One is essential, another is non-essential. Mammals can only synthesize the non-essential amino acids, but the essential ones they must have from their diet. Now, the dietary amino acids are neither stored for future use like lipids nor are excreted, but they are removed. How they are removed? The alpha amino groups from the amino acids are removed and the resulting carbon skeleton enter the TCA cycle. Now, the first stage of protein metabolism is proteolysis. Proteolysis means protein degradation. I will mainly focus on proteolysis of dietary proteins. Now, dietary proteins are hydrolyzed into peptides by different enzymes secreted by the stomach, pancreas, and lumen of the small intestine. Protein digestion begins in the stomach by the enzyme known as pepsin. Pepsin is secreted as its inactive precursor, which is also known as zymogen, and it is secreted from pepsinogen, which is further activated into pepsin. What is the function of pepsin? Pepsin degrades the protein into peptides and few free amino acids. The degradation process continues in the lumen of the small intestine by the various enzymes secreted by the pancreas, namely trypsin, chymotrypsin, carboxypeptidase A and B, and elastase. These enzymes are also secreted from their inactive precursors like trypsinogen, chymotrypsinogen, procarboxypeptidase, and proelastase. Now, the protein digestion is ultimately completed in the lumen of the intestine by an enzyme present there only, which is known as leucine aminopeptidase. Now, this enzyme cleaves the protein from the amino terminal end. In this diagram, we can see that protein digestion takes place in the lumen of the intestine where it is completed, and after the digestion is completed, it is absorbed in the intestinal mucosa. After that, it is transported to various tissues like liver via the blood. I will discuss on flow sheet of amino acid oxidation. That is, there are 20 different multi-enzyme sequences for 20 different naturally occurring amino acids. All of them ultimately enter the TCA cycle through acetyl-CoA, acetoacetyl-CoA, or other TCA cycle intermediates. In this particular diagram, we can see that how the different amino acids enter the Krebs cycle or TCA cycle through either acetyl-CoA or acetoacetyl-CoA or other TCA cycle intermediates. I can give you one example, such as alanine, tryptophan, cysteine, glycine, serine. They are first converted to pyruvate and then to acetyl-CoA, and then the acetyl-CoA enters the TCA cycle. Like this, various amino acids have their specific routes. Not all the carbons of all the 20 different amino acids enter the TCA cycle. Some are lost by decarboxylation reaction. What is decarboxylation reaction? As the name suggests, it results in the release of carbon dioxide from alpha amino acid. Here is shown the particular reaction. From alpha amino acid, carbon dioxide is released, resulting in the formation of primary amine. The enzyme catalyzing this type of reaction is called amino acid decarboxylase, and the coenzyme required is PLP or pyridoxal phosphate. As for example, arginine is decarboxylated to agmatine, which is a primary amine, and the coenzyme required is PLP, and the enzyme is arginine decarboxylase. Now, most of the amino acid catabolism begins with the removal of alpha amino groups. So, I will now discuss the two major pathways by which these alpha amino groups are removed. These are transamination and deamination. I will first discuss about transamination as it occurs first in the sequence. Now, to generalize the reaction, we can see here that from an alpha amino acid, alpha amino group is transferred to an acceptor alpha keto acid called alpha ketoglutarate, which after accepting the amino group is converted to glutamate and another alpha keto acid. Now these transamination reactions are catalyzed by aminotransferases or transaminases, and the coenzyme required is PLP. Now here it is shown the structure of pyridoxal 5' phosphate or PLP and its precursor, which is a vitamin B6 or pyridoxine. Now if we go into the details of transamination reaction, we can see that it occurs through various steps. The first step is the formation of internal albumin. Now internal albumin formation occurs between PLP and the enzyme, and this occurs when the amino acid substrate is absent. What is this internal albumin? It is the shift base linkage between the aldehyde carbon of PLP with the epsilon NH2 group of lysine at the active site of the enzyme. The next step is the conversion of internal albumin to external albumin. Now how does this happen? The epsilon amino group of lysine at the active site of the enzyme is replaced by the alpha amino group of the amino acid and hence results in the formation of external albumin. This external albumin is connected with the enzyme through multiple non-covalent interactions. Now here it is summarized that how internal albumin is converted to external albumin. The next step is the conversion of external albumin to a quinonoid intermediate. How does this happen? This happens by deprotonation from the alpha carbon atom, as we can see in this diagram. From the quinonoid intermediate, ketimine formation occurs by reprotonation at the aldehyde carbon. Next, the ketimine is hydrolyzed into pyridoxamine phosphate and alpha keto acid. Now this constitutes only the half of transamination reaction. So to summarize the half of this reaction, it is the reaction between amino acid 1 and enzyme bound PLP to generate alpha keto acid 1 and enzyme bound PMP. Now the second half of the reaction is just the opposite of the above pathway. To summarize the second half, it is the reaction between alpha keto acid 2 and enzyme bound PMP to generate amino acid 2 and enzyme bound PLP. The sum of these partial reactions is the reaction between amino acid 1 and alpha keto acid 2 to generate amino acid 2 and alpha keto acid 1. Now to exemplify, alanine is transaminated by alanine aminotransferase. How it is transaminated? Now amino group from alanine is transferred to alpha ketoglutarate, resulting in the formation of glutamate and pyruvate. The coenzyme required, I have told already that it is PLP. Now after glutamate is being formed through various transamination reactions, it is oxidatively deaminated to alpha ketoglutarate and the enzyme present is L-glutamate dehydrogenase. The coenzyme required by this enzyme is either NAD+ or NADP+. This reaction, as we can see in this diagram, occurs through an unstable intermediate. The first step is the dehydrogenation and the second step is the hydrolysis of the shift base linkage. The enzyme catalyzing this reaction is glutamate dehydrogenase. Now many organisms have other flavin linked enzymes called L-amino acid oxidase and D-amino acid oxidase that also catalyzes oxidative deamination. Now first I will discuss how L-amino acid oxidase deaminates. L-amino acid oxidase, as the name suggests, is specific for L-amino acid and it is converted to an alpha keto acid through the formation of an unstable intermediate. The coenzyme required here, as we can see, that FMN, which is flavin mononucleotide. After the reaction is complete, FMNH2 is generated, which can be reoxidized to generate FMN and H2O2. Now H2O2 is really toxic for the cell. So it must be detoxified by catalase. Now similarly, D-amino acid oxidase is specific for D-amino acids, and the reaction proceeds similarly through an unstable intermediate, which is shown in third bracket here. The coenzyme used is FAD rather than FMN. FAD is flavin adenine dinucleotide. It is converted after the reaction to FADH2, which is again oxidized to regenerate FAD and H2O2, and this H2O2 is again toxic, which must be detoxified by catalase. Now most of the amino acids are deaminated by this process, but there are two amino acids, L-serine and L-threonine, that can be directly deaminated with the release of ammonia. The enzymes catalyzing these reactions are dehydratase. Now if we see this particular reaction, L-serine is converted to pyruvate with the release of ammonia, and the enzyme catalyzing this reaction is L-serine dehydratase, and the coenzyme is PLP. Similarly, L-threonine is converted to alpha-ketobutyrate with the release of ammonia, and the enzyme is L-threonine dehydratase, and the coenzyme is PLP. The sum of the reactions catalyzed by aminotransferases and L-glutamate dehydrogenase is as follows. Alpha amino acid reacts with NAD+ or NADP+, resulting in the production of alpha keto acid, ammonia, NADH, H+ or NADPH. Now to summarize, the transamination and deamination reaction resulting in the formation of ammonia, and which is ultimately converted to urea and it is then excreted. Let me explain this diagram to you. From the alpha amino acid, amino group is transferred to alpha ketoglutarate, which after accepting the amino group is converted to glutamate, which is then deaminated with the release of ammonia utilizing the coenzyme NAD+. This ammonia is then converted to urea, which is then excreted. Now nitrogen can also be transported to the liver by another route in the form of glutamine from glutamate, catalyzed by glutamine synthetase in an ATP dependent manner. From this glutamine, ammonia is released by the enzyme glutaminase. This ammonia can again be converted to urea in the liver, which is then excreted. These are the pathways by which the alpha amino groups are removed from the amino acid, and the resulting carbon skeleton then enter the TCA cycle by 5 major pathways, that is through acetyl-CoA alpha ketoglutarate, oxaloacetate succinate fumarate. Now of these 5 pathways, the most important one is through acetyl-CoA. Now here we can see that the different amino acids which enter the TCA cycle via pyruvic acid, which is then decarboxylated to form acetyl-CoA, which ultimately enters the TCA cycle. After entering the TCA cycle, through the intermediacy of electron transport and oxidative phosphorylation, it is ultimately converted to carbon dioxide and water, and the free energy is conserved in the form of ATP. Now this is all for today's lecture. In the next lecture, we shall continue on some other aspects of protein metabolism. Thank you.